Hello, and so we are back with our quick review of the structure of DNA and then talking about DNA replication. So on slide 59, we had a blank diagram that you can label. Now, some of these things are pretty obvious, like you should be able to notice the shape of the circle, the phosphate matches. If we were to go back to this diagram, you can see the phosphates are circles, and the sugar, the deoxyribose, is the pentagon. So the deoxyribose would be over here, and there are duplicates over here. So just to practice, here's our phosphate, and then here's our deoxyribose. Now, I left some of them blank right now, so we can go over them. So here, this one is labeled adenine. And if this one is labeled adenine, then that makes this one over here thymine. Now, I will tell you, there is a mistake on this diagram, and I didn't notice it when I first chose it, but then when I noticed it, I wanted to keep it because I wanted to let you know and see if you could figure out there is a problem with this DNA strand, and I want you to see if you can figure it out. So the dotted lines are going to be our hydrogen bonds all the way down in our DNA molecule, our double strand. If this is adenine, then the other one should be thymine. If this is guanine, then this should be cytosine. Notice the A and the G are longer because they're the ones that have the two rings. Now, if this is guanine, then so is this one down below here. And then there, I just need to move the cytosine over, and that would be it. So you can keep looking at this and seeing if you can find the mistake. Let's talk about how we can then replicate our DNA. Here's another practice one if you'd like. You guys can go over that in class if you'd like. So for DNA replication, let me go ahead and present this so you guys can see a bigger view of it. So with DNA replication, one thing we need to realize is that there are proteins, and there are a lot of proteins, and we're just going to go over a couple. There are proteins, mainly enzymes, that carry out the process of copying DNA. One of the first things we need to do when we need to copy DNA, and remember this is happening as part of that S phase of the cell cycle. When you unzip the DNA helix, notice here's our GIF up here. That's what's happening. We're going to unzip the DNA helix, the double helix. There's a protein and enzyme that's going to do that. And as it does that, we're going to call that the replication fork. And where it starts is the origin of replication. Now, that's going to happen in both directions, although this diagram only shows it happening on one side. But just like a zipper, we're going to unzip, but not like a zipper. You're going to unzip a zipper at both ends. So it would be like opening both sides of the DNA. You'd have one over here and one over here, a replication fork. Now that enzyme is going to break the hydrogen bonds that holds the two strands together. Now, what this GIF does not show is that there are nucleotides in the nucleus that are just floating around. We have free nucleotides that comes from the foods that we eat. If you ate an apple today, then the apple has DNA, and we break down the DNA. And so we have nucleotides in our cells that we can use to build new DNA. So what we're going to see is that these nucleotides floating around, they can pair with the nucleotides already there. We can bond using hydrogen bonds. And then there's going to be another enzyme. And this enzyme, I want you to know the name of. The enzyme is called DNA polymerase. And that DNA polymerase is going to form new covalent bonds along the backbone. Almost like glue, it's going to glue the phosphate and deoxyribose together. Now that name comes from, and this is straight from your book does a good job with this, DNA polymerase, the name comes from the fact that you are making a DNA polymer Remember, the polymer is the many linked chain for DNA, and then A stands for an enzyme. So a DNA polymerase is the enzyme that makes the polymer. Oh, sorry. Now, so what you end up having from a holistic view, if we were kind of look backward, not backwards, from far away, what you end up having is the original DNA strand, and you're going to unzip it on one side, and for each of the old original strands, you're going to build a new strand using the old strand as a template. And so as we do that, they're using the yellow to represent the new strands. What you'd end up having is two new double helix molecules, each one with one original purple strand and one new yellow. Now, of course, they're not different colors, but you understand they're just making a color coding. We call this type of replication semi-conservative because you've got one old and one new. And so you're conserving one of the original strands for each side. That's kind of where they came up with that name. So there would be our kind of our basic rundown of the process. Unzip the DNA, match the new ones, and then glue the backbone together so you get two new helices. And so here it is on a more visual process. So here we have the parent DNA, the old original DNA double helix. And now this diagram is showing a lot more of the proteins, but don't worry. What I've done is included, my apologies, included the steps that I want you to know in the boxes. So we've got our protein that's unzipping, that's our replication fork. It's splitting up the old original parent DNA. And one strand, you'll notice, is building it using one DNA polymerase. And here would be our free nucleotides that are pairing up because they match. They form those hydrogen bonds. And then the DNA polymerase is forming covalent bonds all along the side. Remember, covalent bonds are ones where they share electrons. So these are much stronger. So that green would represent the phosphate deoxyribose, and you're making that connection, that covalent bond between them. So this is happening on each side. You have one DNA polymerase over here. We have another DNA polymerase on this side. And so that would be a DNA replication in a nutshell. Now, there are going to be a couple of videos that are going to be super helpful for you to watch. So I'm going to let you guys do that as a class, and then you're going to do a Jamboard, which is going to help practice all of these different processes. So thanks for listening, and have a good day.